Hi folks, welcome back. We're going to finish up this chapter looking at primarily lysosomes, so let's get going. Start off pretty general. Lysosome is an organelle of the endomembrane system, and it contains digestive enzymes. It's capable of degrading all the major classes of biological macromolecules, so lipids, carbohydrates, nucleic acids, proteins as far as we're concerned. These lysosomes contain acid phosphatase and several other hydrolytic enzymes that all work at a low pH, low being lower than cytosolic pH. The organelle varies in size and shape, usually around a half micrometer in diameter, and they're bounded by a single membrane. The luminal side of the membrane, so that's the inside of the lysosome, is coated with glycoprotein, and that protects the membrane from degradation. Otherwise, it would self-digest. The lysosome maintains an acidic environment inside the lumen, usually around pH 4 to 5, and that's due to the action of ATP-dependent proton pumps, like you can see here. And here we pump protons from the cytosol into the lumen of the organelle, and that lowers the pH significantly, 2 orders of magnitude from 7 to 5. It's a 100-fold reduction in 100-fold increase in proton concentration. There are numerous enzymes in the lumen here, like we say, and they are all acid hydrolases. That means they work at low pH and they perform hydrolytic chemistry. So that's what's in there. That's what the organelle does. So what I want to focus on now is how do you get lysosomal proteins into the lysosome? So how do we traffic soluble lysosomal proteins? The soluble part is the acid hydrolases, so they're inside the lumen, free floating. We're not talking about endosomal. We're not talking about lysosomal proteins in the membrane of the lysosome. Okay, so the trafficking of soluble lysosomal proteins is dependent on mannose 6-phosphate addition. So that's the addition of a phosphate to the 6-carbon of mannose, and that happens in the cis-Golgi network. And it's dependent on this enzyme here, N-acetylglucosamine. That's the glucnec there, N-acetylglucosamine phosphotransferase. So is what this enzyme does in the cis-Golgi network is that it binds to a lysosomal protein here. So this is going to be an acid hydrolase. It's going to end up in the lumen of the lysosome. It recognizes the glycosylation that's occurred on the lysosomal protein, and the N-acetylglucosamine phosphotransferase binds an activated phosphorylated N-acetylglucosamine. And it adds that to some sugars which have been added to the lysosomal protein in the ER or the cis-Golgi network. That N-acetylglucosamine with those phosphates is added to one of these mannoses on the oligosaccharide that's been added to the lysosomal protein. And so that was done in the ER or the cis-Golgi network itself. Then there is an enzyme. So there is the oligosaccharide of the lysosomal protein with the phosphate added to the 6-carbon, and there's the N-acetylglucosamine. And so there's an enzyme called phosphodiesterase, and that removes the N-acetylglucosamine, that's the little red square there, to leave phosphorylated mannose 6. So that is mannose, that little residue, that little hexagon there, that little blue one on the end, that is mannose 6-phosphate, and that mannose 6-phosphate there is what will target the this lysosomal protein ultimately to lysosome. So let's take a look at that process. So looking kind of complicated, lots of things going on here. So we'll break it down, and we're going to start down here in the trans-Golgi network, and then we'll look to see how we end up getting this lysosomal protein here all the way into a lysosome, and then we'll talk about some other stuff that's going on up here towards the end. Okay, so in the trans-Golgi network, there are some receptors called mannose 6-phosphate receptors. That's this little kind of orange hook thing here. So the mannose 6-phosphate will bind to mannose 6. So if there's anything with a mannose 6-phosphate on it, it will be bound by the mannose 6-phosphate receptor. And so this lysosomal protein has a mannose 6-phosphate, and so it's bound by the mannose 6 receptor. And then we're going to have the formation of a clathrin-coated vesicle. And so we get budding of the vesicle, and the membrane of the vesicle has the mannose-6-phosphate, the little orange hook, and that's bound to the mannose-6-phosphate of the lysosomal protein that's going to end up going to the lysosome. Then we have uncoating of the clathrin triskelions that we talked about before in the AP complex proteins, and they will be recycled and used to form new clathrin-coated vesicles. So we're left with an uncoated transport vesicle, and that associates and fuses with a late endosome with a low pH. And the low pH, again, as we've mentioned before, that causes the mannose-6-phosphate, which the mannose-6-phosphate receptor, to let go of the mannose-6-phosphate on the lysosomal protein. So that's dissociation of the receptor and the mannose-6-phosphate on the lysosomal protein. And then that means we can end up with the mature lysosomal protein in a lysosome. But we then have the problem of we've got to get this receptor back to the trans-Golgi network. So again, we can form a coated vesicle, and that will either send the mannose-6-phosphate receptor back to the trans-Golgi network, or we can actually send the mannose-6-phosphate receptor to the cell surface. So why would we want to send the mannose-6-phosphate receptor to the cell surface? Well, bear in mind that when we produce vesicles, just vesicles that just bud from the trans-Golgi network that are involved in constitutive secretion, sometimes proteins get carried along unwittingly, if you like. They're just bundled up, a piece of membrane gets pinched off, and you've got a lysosomal protein in there that ends up outside the cell. But if we've got mannose-6 receptors, mannose-6-phosphate receptors on the cell surface, any of those lysosomal proteins that get secreted accidentally can bind to the mannose-6-phosphate receptor, and then we can have clathrin-dependent receptor-mediated endocytosis. And so we talked about this before. We have a vesicle form, we have a coated vesicle which then uncoats, fuses with a late endosome, and then it's the same process for either recycling the mannose-6-phosphate receptor to the trans-Golgi network or sending it out to the cell surface, and we end up with our lysosomal protein in a mature endosome. So that's a neat way of how you can get lysosomal proteins from the ER, from the ER into the cis-Golgi, trans-Golgi, and then ultimately into the lysosomes. So let's just finish up talking about lysosomes in general and degradation of lysosomal contents. So there are really kind of two kinds of lysosomes. There are heterophagic lysosomes, and they contain material which was brought from outside the cell. And then we have those lysosomes which contain material which was engulfed within the cell, and those are called autophagic lysosomes. So we can have two types of lysosome, one which is based on autophagy, or we could bring in material from the cell membrane, and that would then be heterophagic as a lysosome. Regardless, if we're going to take some cytoplasm or an organelle that needs to be broken down, a cup-shaped membrane structure starts to form around the cytoplasm or the structure that needs to be engulfed, and then we keep adding new membrane to this cup. There are a number of proteins involved in recognizing the structure to be engulfed, and eventually we have a double membrane that forms around the structure or the cytoplasm to be degraded, and then this vesicle fuses with a lysosome, and then we can break down the components of the lysosome. And here we're looking at the example of amino acids can be exported into the cytosol. You can just do this with pieces of cytoplasm as well. If you need to recycle cytoplasmic components, you can just engulf a small piece of cytoplasm and fuse that vesicle with the lysosome and break whatever material is in that down and release the components into the cytosol for reuse. So that brings us to the end of chapter 12. Lots of stuff going on here, a lot about trafficking, glycosylation, and we're going to take also a look this week at chapter 16, which is about nuclear structure and function, but it's pretty shallow, and there's a lot of stuff there which is more about genetics, and so I'll be leaving that alone for the geneticists to deal with. I'll see you soon when we're talking about chapter 16. Bye for now.